So I'm going to talk about octopamine-mediated signaling and metabolism in Drosophila aging brain. But first, let me just I will just make a short introduction about noradrenergic signaling in astrocytes and metabolism. So, as you probably know, astrocytes are a subtype of glial cells that are important to give metabolic, homeostatic, and structural support to the neurons. And astrocytes are considered to be the main target of noradrenaline. And after binding of noradrenaline to adrenergic G-protein-coupled receptors, noradrenaline causes changes in intracellular calcium and cyclic AMP signaling pathways. So, in astrocytes, noradrenergic signaling is considered to increase extracellular glucose uptake and glycogen degradation, and also lactate production in a process called aerobic glycolysis. This lactate can be then transported to neurons transferred to neurons by transporters, where it can be used as a fuel in oxidative metabolism to address the fast energy demands in the brain. So it's well known that noradrenaline has important role in many cognitive functions, for example, learning, memory formation, attention. And during aging and neurodegeneration, brain glucose utilization is reduced, and we want to see if malfunction of noradrenergic system is somehow connected to these changes. Because, as I said before, noradrenergic system controls the brain metabolism. So for this, we chose Drosophila as a brain aging model. So in Drosophila, octopamine is a homologue of noradrenaline with similar roles. It modulates metabolism, behavior, learning, memory formation, etc. And it also binds to adrenergic GPCR receptors, which are adrenergic-like receptors, and binding of octopamine to these receptors also causes changes in intracellular calcium and cyclic AMP levels. So in our Drosophila model, we observed neurodegenerative vacuoles in Drosophila aging brain, and also we observed reduced locomotion behavior of this age Drosophila, which was tested with climbing assay. So our aim was to determine if aging affect octopaminergic signaling and metabolism in Drosophila aging brain. So for this, we designed a fly that expressed genetically encoded nanosensors for calcium, cyclic AMP, glucose, and lactate in different cell lines, different cell types of the brain by using a 4UAS system. And for this, we had two different cell lines, different Drosophila lines. One line is carrying a cell-specific promoter and transcription factor GAL4, and we crossed this line with another line which is carrying a gene of interest, in our case sensors, after under control of upstream activating sequence, UAS. And in F1 generation, this GAL4 binds to UAS sequence, and we have expressed our sensor in specific cell lines. So after that, we aged our Drosophila, and we dissect the brain and did the ex vivo brain imaging with confocal microscopy upon octopamine stimulation. So here are our results. Here in these pictures, you can see expression pattern of calcium sensor expressed in astrocytes, cortex glia, which is another type of glia in Drosophila, and in neurons. And as you can see here, octopamine increased the intracellular calcium levels in all 3 cell types. And interestingly, these signals was reduced or even absent in age brain. So this kind of implicate age-related impairment of calcium signaling. So another thing was measurement of cyclic AMP signals, and as you can see, octopamine increases cyclic AMP only in neurons, and we didn't detect any differences between young and age brains. But interestingly, octopamine didn't increase cyclic AMP levels in glial cells, and we were interested why was that so. So we also stimulated the brains with forskolin, which is activator of adenylyl cyclase, and the same as octopamine, also forskolin increased cyclic AMP levels only in neurons, but not in astrocytes. And then we calculated the basal level of this of cyclic AMP in these 3 cell types, and you can see that this basal level was much higher in glial cells. So that may be the reason, because this sensor is operated in relatively narrow concentration range. So this may be the reason. So the sensor was already, you know, the concentration of cyclic AMP was too high for sensor to detect it. But yeah, that's maybe one reason, but also some other reasons can be. So another thing was that we stimulated the brain, which expressed glucose nanosensor, and octopamine increased intracellular glucose levels in astrocytes, but not in neurons. And this suggests glucose uptake in astrocytes, but this signal increase was indeed reduced in age brains. And another thing was to stimulate brains which express lactate sensor, and octopamine increased intracellular lactate levels only in neurons. And this indicates activation of aerobic glycolysis only in neurons, which was, again, really interesting result. And we didn't detect any differences between young and age brains. So then we also exposed brains to elevated concentration of glucose, and you can see that in both cell types, glucose increased intracellular glucose levels, which indicates that both cell types can uptake glucose, but this uptake was significantly reduced in age brains. So to conclude, octopamine triggers intracellular calcium signals in astrocytes, cortex glia, and neurons, but activates cyclic AMP signals only in neurons. Neurons, but not glial cells, exhibit octopamine-induced aerobic glycolysis in Drosophila brains. And in aged Drosophila brains, there is an impairment in octopaminergic regulation of calcium signaling and glucose uptake, as well as reduced delivery of glucose to neurons. So this together may contribute to age-related neurodegeneration and locomotion behavior deficit. So to the end, I would like to thank my supervisor, Nina Vardian, and Anne-Marie Horvat, who also did some of the experience, and all my colleagues at the Institute. And of course, thank you for attention.